Hello everyone, my name is Taylor Hulahan and I'm an MD PhD student in the Angel Lab at the Medical University of South Carolina. Today it's my pleasure to be talking to you about my project titled Investigating the Fibular Collagen Proteomic Metastatic Niche in Axillary Lymph Nodes of Invasive Breast Cancer Patients. I'll begin this presentation with some background on fibular collagens in breast cancer, followed by our mass spectrometry tissue imaging workflow. Then I'll go into our study on an invasive breast cancer cohort from generational South Carolina women, followed by our investigation of the axillary lymph node metastatic niche in triple-negative breast cancer. And finally, I'll leave you with some concluding thoughts. I want to start by highlighting that 36% of diagnoses are made after breast cancer has advanced beyond the localized stage. Even spread to regional areas such as the axillary lymph nodes decreases 5-year survival by 13%. Even spread to regional areas such as the axillary lymph nodes decreases survival by 13%, which we can see here with this graph shown below. In the Angel Lab, we study the spatial organization of extracellular matrix proteins, specifically collagens, throughout breast cancer progression using spatial proteomics techniques such as mass spectrometry tissue imaging. So you might be wondering why we study collagen in breast cancer. Well, collagen has been demonstrated to have translational value in breast cancer. First, in the screening sphere, increased collagen composition has been linked to increased mammographic density. Second, collagen alignment has been associated with survival outcomes. Here we see tumor-associated collagen signature 3, or TAX3, which is defined by these perpendicular collagen fiber alignment to the tumor border, as being associated with worse patient outcomes in breast cancer. Despite these well-understood structural alterations to collagen, we know nothing about its molecular changes throughout breast cancer progression, really highlighting the need for our spatial proteomic studies. In the therapeutic sphere, targeting collagen has been shown to have potential, highlighted in this example of an anti-lysyl oxidase-like antibody, which was found to disrupt invasion through altering collagen fiber organization. Notably, the class of fibular-forming collagens are the most abundant component of the interstitial matrix in the normal breast microenvironment and are actually elevated in breast cancer compared to normal breast tissue. Fibular collagens are characterized by these long interrupted triple helical domains shown in light gray here. Within the triple helical domain, glycine XY is a common motif, where X is most often proline and Y is most often hydroxylated proline. Hydroxylation of proline residues is really crucial in modulating both structure and function of collagen. Hydrogen bonding between strands of collagen increase its structural stability. Hype or hydroxylated proline residues constitute important cell binding domains that are crucial in ECM recognition, signaling, protein binding, and ECM remodeling. An example of this is collagen integrin binding. Notably, hydroxylation of a proline residue within its binding motif increases collagen's affinity for integrin receptors. This impacts how the cells move through the matrix as well as its cell signaling. While most contemporary literature has focused on collagen composition within the primary tumor microenvironment, this study highlighted gene expression differences between the metastatic and primary tumor sites, finding 3 collagen transcripts to be downregulated in the axillary lymph node metastases. This showcases the need for a proteomic investigation of collagens within the axillary lymph node metastases and further understanding of their molecular alterations. Our study aims to investigate the spatial regulation of the collagen proteome within the axillary lymph node metastatic niche. First, by investigating ancestry-dependent stromal variations, and second, through spatially defining the metastatic ECM niche in triple-negative breast cancer. Our mass spectrometry tissue imaging workflow begins with formalin-fixed paraffin-embedded slides. These first undergo N-glycan imaging in collaboration with Dr. Drake's lab and his graduate student Elizabeth Wallace. H&E staining is then performed, followed by histological evaluation by Dr. Laura Spruill. Then we perform collagen imaging, followed by liquid chromatography tandem mass spectrometry for peptide identification. In mass spectrometry tissue imaging, the tissue is sampled by a laser at discrete X and Y coordinates, ionizing peptides from each location. Each X and Y coordinate represents a pixel of data and reports several thousand peaks at this location. Each one of these peaks can then be mapped as an intensity across the tissue, producing a spatial heat map of that peptide's intensity relative to the histology. It's important to note that the spatial distribution of these peptides can then be assessed in relation to pathological annotations. I'm highlighting an example here where we see 3 different peptides with unique spatial distribution across the sample. While some of these peptides have high intensity of expression within the invasive breast cancer region, and others having higher intensity outside this invasive breast cancer region. In the first part of this talk, I will discuss our study of an invasive breast cancer cohort from 31 generational South Carolina women. Before I get into the data, I think it's important to address why studying breast cancer and race in South Carolina is so crucial. While nationally there is a huge disparity in breast cancer mortality rates between Black and white women, it's only exacerbated in South Carolina, where there's a 42% increase in breast cancer mortality between Black and white women. In considering the axillary lympho-metastatic rates, it's important to note that premenopausal Black women have higher rates of lymph node involvement than white women. Our cohort for this study consisted of 31 women from South Carolina within zip codes considered to be a part of the Sea Islanders community. This community has been demonstrated to have a high proportion of West African ancestry with low European admixture, which is associated with more aggressive breast cancers. Tumors for this study were classified as various histological types and hormone receptor statuses. Within this study, multiple cores were taken from primary tumor, normal adjacent tissue, normal lymph nodes, and metastatic lymph nodes to create 3 tissue microarrays. Within the primary tumor site, 3 different post-translationally modified peptides mapping to the alpha-1 chain of collagen type 3 were shown to have different intensity profiles between Black and white patients within the cohort. In related literature, elevated Col3A1 has been associated with increased cancer stemness, specifically in African American patients, but also with tumor cell dormancy. These results really highlight a need to investigate the molecular regulation of the alpha-1 chain of collagen type 3 at a proteomic level. 2 additional peptides were found to have decreased intensities in Black patients compared to white patients within this cohort. One peptide was putatively identified to be from vimentin, and the other from the alpha-1 chain of collagen type 6. Increased vimentin has been associated with epithelial mesenchymal transitions, whereas decreased Col6A1 has been associated with poor survival outcomes within the literature. Within normal lymph node specimens, 153 peptides were discovered to have distinct intensity profiles between Black and white patients. 3 are highlighted here. Peptides 1020 and 1577 were putatively identified as from Col6A3, and peptide 1562 as from Col14A1. Interestingly, peptide 1562 was not only identified as having differential intensities of normal lymph nodes between Black and white patients, but was also found to have increased intensity in benign lymph nodes compared to metastatic lymph nodes within the white patient subset. It's interesting to note that Col14A1 has been shown to be transcriptionally downregulated with increasing tumor grade. Within axillary lymph node metastases, 46 peptides were discovered to have distinct intensity profiles between Black and white patients. Peptide 843 was putatively identified to be from the alpha-1 chain of collagen type 1, while peptide 1263 was mapped to the alpha-3 chain of collagen type 6. In comparing specimen types, peptide 843 was additionally found to have increased intensity patterns within the axillary lymph node mets compared to the normal lymph nodes within the Black patient subset. Col1A1 expression has been shown to be important in invasion and migration of breast cancer cells in knockdown studies. Within this first study, we found ancestry-dependent stromal proteomic variations in the primary tumor, axillary lymph node metastases, and benign lymph nodes. This led to our subsequent investigation of the axillary lymph node metastatic niche in triple-negative breast cancer patients. Within this section, I'll be showing some mass spectrometry tissue images where intensities of different peptides will be mapped across the tissue, with red indicating high intensity and blue indicating low intensity. The ancestry-dependent stromal proteomic differences seen in the first study led us to investigate triple-negative breast cancer, as triple-negative breast cancer disproportionately impacts Black women. While not the most prevalent metastatic site in triple-negative breast cancer, axillary lymph node metastases and spread to nearby areas decreases 5-year survival rates by 26%. An overall survival curve is shown here from a study of 145 TNBC patients that were sampled from the Cancer Hospital of Harbin in Harbin, China. Perhaps it is not surprising that more lymph node metastases were found to be associated with poorer survival outcomes. This study reiterates the importance of lymph node metastases as prognosticators, but also draws attention to the need for more racially diverse studies. In our preliminary cohort from 5 triple-negative breast cancer patients, we acquired primary tumor, metastatic lymph node, and benign lymph node specimens for each patient. To capture an overall view of collagen fiber maturity, we performed Hurahan staining. In this control slide of pediatric aortic valve, blue indicates immature collagen fibers, while red reports mature collagen fibers. In all 3 specimen types, we found a predominance of red staining, which seems to suggest maturity of collagen fiber networks. The literature reports an association between high red staining and increased type 1 collagen fiber abundance. And it's interesting to note, as I discuss our imaging mass spectrometry data in the following slides. Now that we've looked at the maturity of collagen fiber networks using Hurahan staining, we'll zoom in to look at the spatial organization of discrete peptides using our ECM-targeted mass spectrometry imaging methodology. Our segmentation analysis of over 135,000 pixels demonstrated 19 unique proteomic clusters, with certain clusters overrepresented within the metastatic lymph nodes. Looking specifically at this dark blue cluster, we can visually appreciate its enrichment in the metastatic lymph nodes. A comparison of average intensity profiles between specimen types of 187 ECM peptide peaks demonstrates a distinct ECM proteomic signature within the metastatic niche. This is nicely recapitulated in an orthogonal sparse partial least squares discriminant analysis, where we see distinct cluster for each specimen type. Here we can visually appreciate the spatial distribution of one of the identified upregulated peptide peaks in the metastatic niche. This peptide was punitively mapped to vimentin. Its highest intensities were within the metastatic lymph nodes, and low intensities of this peak can be noted in the primary tumor, while almost no peak intensity was detected within the benign lymph node. Receiver operator curve analysis, or ROC analysis, from spectral data of one patient demonstrates the ability of this peptide to discriminate between metastatic and benign lymph nodes. But how does this relate back to the literature? Vimentin is a type 3 intermediate filament that has been demonstrated to be upregulated in metastatic breast cancer and associate with poor patient outcomes. Vimentin has also been linked to the suppression of epithelial features and increased cell migration. In its extracellular forms, it's been associated with human diseases. So all this to say that our data seems to coincide nicely with literature on vimentin. Given the importance of collagen in breast cancer progression, as I've highlighted throughout this talk, I wanted to showcase a peptide punitively identified from the alpha-2 chain of collagen type 1 that contained 2 hydroxylated proline sites. This peptide demonstrated increased intensities within the metastatic lymph nodes and lower intensities within the primary tumor site. ROC analysis depicted its ability to discriminate between benign and metastatic lymph nodes. In a previous mass spectrometry imaging study, another alpha-2 chain collagen type 1 peptide was identified to be elevated in triple-negative breast cancer compared to benign breast tissue. Additionally, the study demonstrated that high COL1A2 transcript levels were associated with poor survival outcomes in breast cancer, which further supports our findings. Using Haravici staining, we characterized the collagen matrix to be predominantly mature within the primary tumor, metastatic site, and benign lymph nodes. Using mass spectrometry tissue imaging, we identified a unique ECM proteomic signature within the axillary lymph node metastases. Within these studies, we discovered ancestry-dependent stromal proteomic variations across the specimen type sampled. We additionally found a unique ECM proteomic signature of axillary lymph node metastases. While pathology is invaluable in understanding invasive breast cancer, supplementing this with molecular alterations throughout disease progression offer important insights into the biology of invasive breast cancer and could lead to novel prognosticators. Future work will focus on validating these findings in a larger patient cohort to establish its prognostic potential. I'd like to thank my lab, as well as the Meta Lab and the Drake Lab at the Medical University of South Carolina and the Proteomics facility, as well as all of our collaborators and of course all the patients that made this study possible, and to you all for listening.